Pharmacology. The second chapter name is Drug Receptor Interactions and Pharmacodynamics, written by Jonah Paris, chapter number 2. Let's start with overview. Pharmacodynamics describe the action of a drug on the body. Most drugs exert effect both beneficial and harmful by interacting with specialized targeted macromolecules called receptors, which are present on or in the cell. The drug receptor complex initiates alterations in biochemical and or molecular activity of cell by a process called single transduction. Now let's we see what is single transduction. Drug act as signal transduction. Drug act as signals and receptor act as signal detectors. A drug is termed an agonist if it binds to a site on a receptor protein and activates it to initiate a series of reactions that ultimately result in a specific intracellular response. Second, messenger or effector molecules are part of the cascade of events that translates agonist binding into a cellular response. You can see the all diagrams here. First receptor, second drug attach bind with receptor and start with active receptor activated, it activates the receptor and then biological response start of drugs. No, let's start with the drug receptor complex. Cells have many different types of receptors, as you know that, each of which is specific for a particular organism and produces a unique response. Cardiac cell membrane, for example, contain beta adrenergic receptor that bind and respond to epinephrine or norepinephrine. Cardiac cells also contain muscarinic receptor that bind and respond to acetylcholine. These two receptor populations dynamically interact to control the heart's vital function. The magnitude of the cellular response is proportional to the number of drug receptor complexes. This concept is conceptually similar to the formation of complexes between enzyme and substrate and shares many common features, such as specificity of the receptor for a given agonist. Although much of this chapter centers on the interaction of drug with specific receptors, it is important to know that not all drugs exert effect by interacting with the receptor. Antacids, for instance, chemically neutralize excess gastric acid, thereby reducing stomach upset. Now let's start receptor states. Receptor exist in at least two states, inactive and active. When it is inactive, it is shown with R, and when it is active, it is shown with R positive, that are in reversible equilibrium with one another, usually favoring the inactive state. Binding of agonists cause the equilibrium to shift from R to R positive to produce a biological effect. Antagonists are drugs that bind to the receptor but do not increase the fractions of R positive, instead stabilizing the fraction of R. Some drugs partial agonists shift the equilibrium from R to R positive, but the fraction of R positive is less than that caused by an agonist. The magnitude of biological effect is directly related to the fraction of R positive. In summary, agonists, antagonists, and partial agonists are examples of molecules or ligands that bind to the activation site on the receptor and can affect the fraction of R positive. Major receptor families. A receptor is defined as a biological molecule to which a drug binds and produces a measurable response. Thus, enzymes, nucleic acids, and structural proteins can act as receptors for a drug or androgenous agonist. However, the richest source of receptors are membrane-bound proteins that transduce extracellular signals into intracellular response. These receptors may be divided into four families: ligand-gated ion channels, G protein-coupled receptors, enzymes linked receptor, intracellular receptors. Generally, hydrophilic ligands interact with receptors that are found on the cell surface. In contrast, hydrophobic ligands with receptors found inside cells, figure 2.2, you can see that here is the figure 2.2. All the intracellular tracts. Next lecture we will discuss about this diagram, inshallah.